Hey everyone, this particular lecture is going to be looking at your textbook chapter 6, and in particular we're going to be looking at enzyme structure and function. So you are going to notice this lecture does have some overlap with our discussion of proteins in chapter 3. It also has overlap with the previous lecture, which was mostly about energy, in particular looking at this thing called activation energy, which is the amount of energy needed to start a chemical reaction. So in order to get ourselves started talking about enzymes and energy, we should talk about what enzymes actually are. So let's take a look at some of the traits of enzymes, okay? The first thing to know, you probably know this already from chapter 3, enzymes are proteins. Now there are other types of things that can operate as enzymes, but generally speaking, if something is called an enzyme, it implies that it is also a protein, okay? And you might remember from chapter 3 as well, that with proteins, their three-dimensional shape, their tertiary structure, determines their function. Okay, so our lovely purple blob here is a representation of an enzyme. Okay, and each of these little circles on our purple blob, that's one of the atoms within our enzyme. So you can see enzymes can be pretty big. Okay, lots and lots of atoms all put together to make a particular shape. A couple other things to know about enzymes. First off, they're defined by their protein nature, and they're also defined by their function, which is to be a catalyst. In other words, they're something that speed up chemical reactions. Now, catalysts can come in a lot of different forms, okay? Anything that speeds up any chemical reaction can be called a catalyst. However, enzyme catalysts have a really specific way, not just of being as a protein, but also a specific way of doing their job, okay? When they speed up their chemical reaction, they do it by making the action, right, that whatever that chemical reaction is, easier to perform. In other words, they lower the activation energy. So you might remember from our last lecture that activation energy, right, is the amount of energy it takes to start a chemical reaction. If you lower that initial investment, it makes the whole reaction easier to accomplish. That is the goal of the enzyme, to lower that initial investment so that you can complete the reaction with less energy overall. Now here's the thing. They do this in a really specific way, based on their three-dimensional shape. Enzymes are specific to the reactions that they will catalyze because they are going to use their three-dimensional shape to do the job. If your reaction doesn't match that three-dimensional shape, the enzyme can't help you. So you need to have a match between the three-dimensional shape of the enzyme and the three-dimensional shape of whatever it is you're acting upon, which is called your reactant, right, for a chemical reaction. Or sometimes it's also called a substrate if you're talking about enzymes in particular. Now, the reason why enzymes can do this particular job of lowering activation energy, okay, based on shape, is because they do it in one very specific way. What they do is they match up their shape, which is called the active site of that enzyme, with one or more of the things that are the starting materials, the reactants, okay? And when you have a reactant in an enzyme, we sometimes refer to that as a substrate. So you can see that word here, substrate. Now, enzymes work a lot like staplers, okay? So if you imagine a stapler for just a moment, right? A stapler has a very specific shape, a very specific size, okay, to do one very specific job. Staplers' shape determines their function. If they didn't have the right shape to hold on to a staple and push it in just the right way, then you wouldn't be able to use that stapler as a stapler, right? That also means you can't use your stapler to put push pins into things, right? They're not the right shape. They won't fit into the stapler. You can't use your stapler to, do something on a computer or something like that, or you shouldn't, right? Because once again, the shape doesn't match the task at hand. Enzymes are the same way. You have to have a match between the shape of the enzyme and the shape of whatever it is that you're accomplishing. Also similar to a stapler, if you imagine our lovely little enzyme here as a stapler and my substrate in here as the staple, the enzyme is not using up its own energy to perform the change, right? So remember, all change, all work takes energy. Enzymes don't use up their own energy when they are lowering the activation energy, when they're making the process easier. Just like staplers don't use their own energy, right? Unless they're one of those electrical ones. But a manual stapler uses your energy from your hand, right? It just makes it easier than trying to have to take a single staple and push it through a piece of paper and then close it with your fingers, right? There's way less energy involved in stapling with a stapler. You take advantage of the shape of the stapler, right? And your own energy to complete the task. That's what enzymes do as well. Also, because of that, right, because they don't use up their own energy and they don't use up their own physical structure, right, they just sort of temporarily use the structure, okay, enzymes can be reused over and over and over again. So just like a stapler, you don't have to throw it away after you've stapled your first chunk of paper, right? You can keep using it over and over and over again, okay? And eventually it wears down, but so do enzymes, okay? Eventually they wear down and break down and stop working. You need to make more. But a stapler, right, allows you to keep performing this action as long as you keep putting in the energy to staple things. Enzymes do the same thing, right? Until you might run out of staples, run out of substrate. So enzymes and staplers actually have quite a lot in common. There is one other thing that they have in common as well, and I'm going to show you this on this picture. You'll see it defined on a future slide, okay? Because they're using their three-dimensional shape to cause a change, that means that their three-dimensional shape, in fact, needs to change, just like the stapler, right? For it to perform the task, it temporarily needs to take a slightly different shape than it was originally, pressing down on the stapler. Enzymes are the same way. They temporarily change their shape in order to permanently alter their substrate. We permanently alter the staple. That temporary change is called a conformational change, and you'll see that in a couple of slides. So once again, a temporary change in the enzyme leads to a permanent change in its substrate or reactants. I'm not concerned that you know these two words, degradation and synthesis, but I did want you to see that it's not all about stapling staples. Sometimes your enzyme is going to be used to take something apart, right, by matching up its active site, its sort of stapler end, if you will, that matches in shape to the substrate, right? So you can stick those two things together. Your enzyme temporarily changes its shape to pull these two things apart. Then it goes back to its normal enzyme shape, and you've got your products, your two new things instead. Conversely, you can take an enzyme, right, with two different things and use it to squish them together. That's what we see over here, a synthesis, a building. In both cases, if you notice, the enzyme's starting shape and ending shape is the same. It changes temporarily in order to force your substrate to become the product, just like your stapler changes its shape. So once again, you temporarily change the shape of the enzyme, okay? That is called a conformational change, okay? And once again, this is why they need to be specific to their substrate, specific to the thing that they're operating on, because you need to be able to have a match to the shape to start with, and you need to be able to change the shape of the thing you're working on. And remember, once again, activation energy, the amount of energy that it takes to get a chemical reaction to happen. This lovely little chart over here is in your textbook, okay? And this is showing you what an enzyme can do for you. So if you figure, if you've got a general amount of energy kicking around and you want to complete a chemical reaction over time, okay, you can either take all this time to get enough energy together, okay, and then say, hey, cool, I've got enough energy together, let's do this thing, and then perform the reaction. Or you can say, hey, you know what, I'm going to use an enzyme instead. I only need this much energy now because the enzyme shape is making this whole process easier, okay, and you'll notice that what winds up happening is the reaction takes less energy, the progress moves a little bit more efficiently, it allows you to get more stuff done. If we look at enzymes in the context of homeostasis, right, we know that our enzyme shape allows it to perform a particular function. It's only going to perform the function on the particular substrates or starting materials that we need it to work on, okay? So in order to get an enzyme to work, you need to give it the right tool. To get a stapler to work, you need to give it staples. Enzymes are the same way. But enzymes are also proteins, which means that their shapes are impacted by the environment. So if you think about it, right, proteins are often held into their tertiary structure by things like hydrogen bonds and interactions between all those amino acids. And hydrogen bonds and interactions between amino acids can actually be changed by extremes in the environment, things like an extreme pH, right? Or a really low or high temperature, or extreme pressure, or like a lot of salt content and stuff like that. What we find is that when you start messing around with the environment, you mess around with how well a machine can work. We know this intuitively. Proteins are the same way. And what we find is that enzymes tend to have a particular environmental range in which they're really, really happy. And you can usually predict that based on what those enzymes are used for and the organism in which they are used, okay? However, there are a couple of patterns that I do want you to be aware of that are fairly uniform. One of those is that of temperature, because temperature is heat, right? And heat is energy. A higher temperature in your system tends to make your enzymes work faster, only because you've already got more energy in the system to begin with, right? And enzymes are making it less energetically costly to do things. So if you've got lots and lots of energy and something that's cheap to do, you can do it faster. However, If your temperature gets too high, right, you can break those hydrogen bonds. You can break the three-dimensional structure or denature. So a temperature that gets too high that breaks those bonds, okay, is actually going to stop the enzyme from working. We can see that sometimes the sensitivity to temperature is really a very subtle difference. So the classic example here, this lovely little cat, okay, you've seen this before with Siamese cats and other types of cats. Sometimes they're called color point because they tend to have a darker shade of fur at their points, right, at their ears, let's say, or at their nose compared to the rest of them. If you were to do a little heat map of this cat, what you would find is that all of the lighter fur is in places that are warmer on the cat, and all of the darker fur is in places that tend to be cooler on the cat, okay? And that is because the enzymes that form the light versus the dark pigments, okay, are more active on the warmer portions, less active on the cooler portions, and so you get that classic color point with the darker ears and the darker nose and all that good stuff. It's kind of a fun thing. Now, if you think about staplers, right, you get to decide when a stapler staples. Enzymes, on the other hand, do not have little hands controlling them. So sometimes you've got to be able to have an enzyme at the ready, right, like having a stapler on your desk, right, and you need to tell it when to turn on. For that, we have things called cofactors, okay? Cofactors work as an on switch, or if you've ever seen something where you need two keys turning at the same time to get it to work, it's sort of the same idea. A cofactor makes the active site the right shape for your substrate, okay? In other words, only in the presence of both the cofactor and the substrate will this enzyme do its job, okay? It turns the enzyme on. The term cofactor can refer to any molecule that does this job, that basically makes the enzyme the right shape in order to work, okay? Sometimes you're going to see this word instead, coenzyme, all right? And all that means is you've got a cofactor that's organic in nature, okay? A lot of our vitamins are either coenzymes on their own, or they're used to build coenzymes. So that's a nice little fact for you to keep in mind. So on switches are great, right? So now you have an enzyme, you can keep it around all the time, but only when you have a cofactor will it turn on. Cool, you can control it. Sometimes, though, you want something that's on all the time, and you want to be able to turn it off when things are getting overloaded, right? So sometimes, rather than having an on switch, your enzyme can actually have an off switch. We refer to the off switches as inhibitors, all right? And inhibitors can work in a couple of different ways. Sometimes they're going to work basically by getting in the way of the enzyme and the substrate. So if you imagine that stapler analogy again, right? You can inhibit the stapler from working by sticking a piece of gum where the staple should come out of the stapler, right? It's going to physically block the part that does the work. That is called a competitive inhibitor. It is a molecule that will sit in the active site of the enzyme and prevent it from actually performing its usual function. Sometimes, though, something even more sort of indirect can happen to turn an enzyme off. These are called non-competitive inhibitors, okay? That's what this picture is over here on the left, okay? So if you've got an enzyme, here's our enzyme here labeled E1, okay? And here's its substrate, object A. You'll see object A fits really nicely into its active site over here, okay? What can happen is that sometimes you might already have enough of whatever this enzyme makes. And they're showing this with multiple steps, but you can even do it with just one. What you want is to be able to turn this enzyme off so that it doesn't continue to make product when you don't need it. So it has a secondary site on it called an allosteric site. You don't need that as a vocabulary word, but it's a nice one to just sort of see this particular picture. The allosteric site matches up with something that can change the shape of the enzyme so that its active site no longer matches the substrate. So what you would have here is a non-competitive inhibitor that when it's in the presence of the enzyme, it says, hey enzyme, change your active site so that the substrate can't fit. These are what are called non-competitive inhibitors. Finally, I did want to show you a couple terms that are in your book in chapter 6, but we're not really going to get to them until chapter 7. Oxidation and reduction, often caused with enzymatic reactions, okay? And they simply refer to the loss or gain of electrons, okay? That's all you need to know about them for now, but I want you to see them because they are in chapter 6, and we will be talking about them later. Here's your list of terms.